Welcome to Section 7 of Immunology. In this section, we'll be discussing major histocompatibility complexes and HLA genes. Let's get started. Before we discuss MHC, it's important that you have a very basic understanding of T cells because these are what interact with MHC. So here's an image of a CD4 positive helper T cell and a CD8 positive cytotoxic T cell. There are three main components that you need to be familiar with. The first one is the T cell receptor. This is an important receptor that all T cells have, and it helps them recognize MHC complexes. We'll talk more about this in a second. The next component is CD3. This is a complex that surrounds the T cell receptor and is responsible for transmitting signals intracellularly. Finally, T cells have either a CD4 surface protein or a CD8 surface protein. So all T cells have a T cell receptor in CD3, but the CD4 or CD8 surface proteins are really what distinguish the T cells from one another. So the T cell receptor binds to MHC, CD3 transmits the signal intracellularly. If the T cell has a CD4 surface protein, it's called a T helper cell. These cells have many functions. I'm not going to discuss them all here, but two important ones to keep in mind are that they stimulate B cells to make antibodies, and they produce cytokines. On the other hand, if the T cell has a CD8 surface protein, it's called a cytotoxic T cell. These directly kill cells, such as cells infected by a virus. All right, with this understanding, let's discuss MHC. MHCs are protein structures found on cell membranes that present antigen fragments to T cell receptors. MHCs are encoded by genes known as HLA genes. There are two important classes of MHC that you should know for step 1, MHC class 1 and MHC class 2. There are some important differences between these two MHCs, so let's talk about them now. Here is the summary chart of major differences between MHC1 and MHC2 molecules. We'll use this chart as an overview throughout the lecture. Let's walk through these points together now. First, there are differences in structure. MHC1 is made of one long chain, shown in dark purple on the left right here, and one short chain, shown in bright pink right here. The long chain is composed entirely of alpha subunits, called alpha 1, 2, and 3. The short chain is composed of a single beta subunit, called beta 2 microglobulin. MHC2, on the other hand, is made up of two chains of equal length. So you can see that one side is purple and one side is pink. One chain has only alpha subunits called alpha 1 and alpha 2, and the other chain has only beta subunits called beta 1 and beta 2. Now let's talk about differences in expression and function. MHC1 is expressed on all nucleated cells, as well as platelets and antigen-presenting cells, or APCs. MHC2 is expressed on antigen-presenting cells only. This is easier to remember if you think about the function of the MHCs. MHC1 presents intracellular antigens, so antigens that are made within the cell itself, and they present these intracellular peptides to cytotoxic T cells, which are responsible for directly killing cells. Let's take a look at an example. This image shows the infected cell right here and a CD8 positive T cell, or cytotoxic T cell, right here. There are some extra details here which we'll discuss in our video on cytotoxic T cells. For now, I just want to point out that the infected cell has MHC1 on its surface, where an antigen is presented to a T cell receptor, or TCR, on a cytotoxic T cell. So remember, MHC1 is on almost all cells, and just in case a healthy cell does become infected, they all have MHC1 so that the cell will be able to be recognized by a CD8 positive T cell and destroyed. Although here we used an example of a viral infection, you should know that cancerous cells can also produce abnormal proteins, which when placed on MHC1 are recognized by CD8 positive T cells. The same happens with donor graft cells. Okay, so from our chart, we covered the expression and function of MHC1. Remember that it's expressed on all nucleated cells, as well as platelets and APCs, and they function to present intracellular antigens to CD8 positive T cells. MHC2 differs in both expression and function. It's expressed only on APCs, and its function is to present extracellular antigens to CD4 positive T cells. Let's take a closer look using another example. So here we can see an antigen-presenting immune cell, such as a macrophage or a dendritic cell. We can also see a CD4 positive T helper cell down here. Notice that once the APC has an antigen on an MHC2 molecule, that the CD4 positive T helper cell binds to the MHC2 complex with its T cell receptor, or TCR. Also notice that CD4 is involved in this interaction. Binding of CD4 is vital for the T cell receptor to produce an intracellular signal. When a T cell binds to MHC2, it can then perform its effector function, which would be to release cytokines and stimulate B cells to make antibodies against this particular antigen. Okay, so now we know that MHC2 are found only on APCs, and their function is to present extracellular antigens to CD4 positive T cells. Also remember that MHC2 binds to CD4 and T cell receptors, whereas MHC1 binds CD8 and T cell receptors. You can remember this by the rule of 8. 1 times 8 equals 8. So MHC1 only interacts with CD8. Also, 2 times 4 equals 8. So MHC2 only interacts with CD4. In other words, just think of the math being equal to 8. 1 times 8 equals 8, and 2 times 4 equals 8. All right, now let's discuss how antigens actually get on to MHC. Let's start with MHC1. Okay, so here we have a picture of a cell. Remember, MHC1 is expressed on all nucleated cells, APCs, and platelets. Also remember that it presents proteins that are produced intracellularly. So here in our image, we have a protein. It's first cleaved by the proteasome into small peptides. The small peptides are transported into the endoplasmic reticulum via the transporter called TAP, which stands for Transporter Associated with Antigen Processing. The peptide fragment is then mounted on the MHC1 molecule within the endoplasmic reticulum, which you can see right here. From here, the MHC1, along with the mounted peptide, goes to the Golgi apparatus, into a secretory vesicle, and then fuses with the cell membrane. So on the surface, we can see that a peptide is mounted on an MHC class 1 molecule right here. A high-yield disorder to be familiar with that's associated with MHC1 is Bayer lymphocyte syndrome. In this condition, there is a mutation in the genes encoding for TAP. As we just learned, TAP is necessary for MHC1 expression, so this results in an absence of MHC class 1. Because MHC1 interacts directly with CD8 positive cytotoxic T cells, this results in impaired cytotoxic T cell activity. Finally, the lymphocyte count is normal, and this is important because it helps distinguish it from other immunodeficiencies. All right, now let's move on to discuss how antigen presentation on MHC class 2 molecules develops. Recall that MHC2 presents extracellular antigens to T cells. So let's look at our diagram here. First, the extracellular pathogen, which we can see right here, is brought into the cell via phagocytosis. Next, the phagosome and lysosome will fuse to form a phagolysosome. Here, the digestive enzymes will break up the pathogen. So notice that here we've labeled the diagram digested pathogen. Okay, now let's take a close look at the endoplasmic reticulum. Here is where the MHC2 protein complex is synthesized. However, it's simultaneously synthesized with many other proteins. So there must be a mechanism in place that prevents unwanted peptides and proteins from binding on the MHC2. This is achieved with the invariant chain, which you can see right here. So notice that the invariant chain blocks the binding site, so nothing binds until the MHC2 meets the proper antigen. Later, the invariant chain is cleaved to form a different version of the invariant chain called class 2 associated invariant chain peptide, or CLIP. CLIP serves the same purpose as the invariant chain, but it's just a version that's easier to remove. Next, vesicles from the endoplasmic reticulum merge with the phagolysosome, and the antigen is swapped with CLIP on the MHC2. So here we can see that CLIP is removed, and the antigen is now loaded onto MHC2. Finally, the phagolysosome merges with the cell membrane to place the MHC2 on the cell membrane, ready to be recognized by a CD4 positive T helper cell. So we can see it on the cell membrane right here. Okay, so now we covered antigen loading for both MHC1 and MHC2. The main points are summarized here on our chart. The last point we want to cover in this lecture is the genetic loci of MHCs. As we mentioned earlier, MHCs are encoded on a gene complex called HLA. Within that gene complex, specific loci code for either MHC1 or MHC2. Let's take a look at this visually. Here we have chromosome 6. The entire HLA gene complex would be this tiny section here, but as we can see, it's blown up on the left. The arrows here are all pointing to specific loci on the gene. MHC1 is encoded on loci HLA-A, HLA-AB, and HLA-AC. MHC2, on the other hand, is encoded on HLA-DP, HLA-DQ, and HLA-DR. Remember, A, B, and C are one letter, so that means they are MHC1, and DP, DQ, and DR are two letters for MHC2. One important thing to mention as well is that these genes are codominant, meaning both the father's and mother's alleles are expressed at the same time. This means that for MHC1, 6 genes are expressed: HLA-A, B, and C from mom, and also from dad. The same goes for HLA-DR, DQ, and DP. The loci of the HLA genes are also highly polymorphic, which just refers to the occurrence of 2 or more alleles at one locus. So for example, on the loci HLA-A, One allele could be denoted as HLA-A1, and a different allele could be denoted as HLA-A2. Both code for the same thing, they are just variants of each other. Some of these variants, or alleles, are associated with disease. Let's briefly mention some of these diseases now. The next two slides will go over disease associations. However, don't memorize this now. Spend time memorizing them when you learn about the actual disease so it has more context. With this in mind, here are the HLA alleles or subtypes you should know for MHC class 1. HLA-A3 is associated with hemochromatosis. HLA-B8 is associated with Addison's disease, myasthenia gravis, and Graves' disease. HLA-B27 is a very important subtype associated with psoriatic arthritis, ankylosing spondylitis, IBD-associated arthritis, and reactive arthritis. Lastly, HLA-C is associated with psoriasis. Remember, all of these are associated with MHC class 1. MHC class 2 HLA alleles also have important associations. Let's briefly run through these, and remember, we will mention these when we cover each disease specifically too. So HLA-DQ has alleles HLA-DQ2 and HLA-DQ8, and these are associated with celiac disease. The rest of the alleles are for HLA-DR. The alleles are HLA-DR2, 3, 4, and 5. So DR2 is associated with multiple sclerosis, hay fever, SLE, and Goodpasture syndrome. DR3 is associated with diabetes mellitus type 1, SLE, Graves' disease, Hashimoto's thyroiditis, and Addison's disease. DR4 is associated with rheumatoid arthritis, diabetes mellitus type 1, and Addison's disease. And finally, HLA-DR5 is associated with Hashimoto's thyroiditis. Okay, now that we've discussed the content, let's review with a question. Researchers are studying the effects of knockout mutations of the gene associated with the invariant chain that is normally loaded onto MHC class 2 molecules. A defect in the invariant chain would most likely result in the presentation of what type of antigens? Okay, so we're asked about the invariant chain. Recall from this image that the MHC2 complex is synthesized in the rough endoplasmic reticulum along with many other proteins. The invariant chain, which we can see right here, normally prevents self-antigens from being loaded onto the MHC2 complex. So if the invariant chain were defective, then the protein synthesized in the rough ER could be loaded onto the MHC2 complex, and the antigen-presenting cell would present self-antigens. So the answer to the question is self-antigens. Obviously, this is problematic because it could result in autoimmune disorders. All right, that should be everything you need to know about major histocompatibility complexes.